what I was asked to do is just talk about where we stand within the genomics of quinoa. I'm excited about this stuff, but I know that it's dry for some people. So I'm going to just try to give you guys a sense of what we're doing, let you know that as much as we say it's an orphan crop, there's research being done on the genetics. We have most of the genetic tools that you would want to use if you want to do marker-assisted selection, if you want to think about sequencing. Those tools are going to be available for you. I'll just show you where we stand in that. And then I also want to make the pitch that it's not just quinoa that we should think about. There's a lot of other crops in Latin America that are certainly worth thinking about. And so I've loaded my slides with a bunch of Amaranth stuff too, because you guys are going to see out in the field, I love Amaranth as well as quinoa. And of course, my interactions with Luz and with Willie and Alejandro Bonifacio and others throughout Latin America, they always bring to me a new crop and say, why don't you work on Papalisa, or why don't you work on Oca or tarweed or something else? And so it's an exciting place to work on these orphan crops of Latin America. You guys know about quinoa. The only thing I would point out to you guys again is that incredible where it's growing, the opportunity to use quinoa in, I'm going to bring that down just a little bit, you guys. It's incredible to use this crop. I still think there's a lot of feedback. I'll try to talk quieter. An incredible crop for where it's being grown. If you think about it in where we live in Utah, there's a big mountain that's right behind our university called Mount Timpanogos. And the students love to climb up to the top of Mount Timpanogos. You can only do it during the summer. Other than that, it has a glacier on the back side of it. But the top of Mount Timpanogos obviously is barren. There's no tree life up there. It got a glacier on the back side of it, and it sits out at about 11,800 feet. Quinoa on the Altiplano in Bolivia, you've got 2 million people subsisting, farmers. They're living on a high plateau with mountains shooting off of that, and the plateau, their valley, is sitting at 12,000, 13,000 feet above sea level. You can't grow much more up there than some of these crops that are ideally suited for these climates. The other thing I would point out to you guys, just so you can think about this, how much rain it gets. That's droughty soils. You think about a place like Oregon, you get that in one rainfall. So this is pretty incredible that you can grow these types of things out there. Here's up on the Altiplano, here's Rick and I, and you guys can see the types of soils conditions that they're using, and then these peaks that are shooting off all the way up to about 22,000, 23,000 feet up there. And this is the high Andean plateau. But again, the droughty soils, frequent frost, pretty incredible ecosystem up there. And yet you can grow really nice crops up here. Here's a variety coming out of Proempa. I'm sure that Wilfredo Rojas will talk more about what they're doing at quinoa, but incredible the progress they've made up there. When you think about this sand, and yet you have these nice varieties that they're developing. They're not just selections; they're varieties through selections off of hybrids, of crosses. So pretty incredible system out there. Just a beautiful place to visit. As quinoa people interested in quinoa, you should contact. I'm sorry, Luz, I'm going to do this to Luz, I'm going to do this. Contact these guys and say, I'd like to come down and visit your area of production. And they take you to places like this, and it's just incredible. Oh, Oregon? That's where you want to go?
Oh. Turn the light off. Someone wanted to take notes in the back, though. So
oh, but you can't
see the pictures.
Ah.
Can't see the pictures. Oh, hang on. Let me try one.
Yeah. It gets ugly. Oh. Yeah, well, okay.
Take notes later.
You go up there, and I always show this to my students in my genetics course, and I said, where is this? And they see this big salt plain back here. And of course, they all think it's Salt Lake, the Great Salt Lake in Utah. And I tell them, no, this is what you can see from the the spaceships, and this is how they mark it. They can see the Salar de Uyuni. It's this massive salt lake. Nothing else is going to be growing up here in these desert environments, but then you can see quinoa, big patches in these cooperative farms, and they're doing well. And so it can not only handle low water, but high salt. About the one thing you're going to grow up here. Here's what corn looks like. Corn not supposed to look like that, you guys, right? The ear is not supposed to come out of the ground, but yet you can grow quinoa up here without any problem at all. I shouldn't say any problem. Certainly, you have to worry about different bugs and different issues up there, but an incredible crop. Another crop I'd like to introduce you guys to is Amaranth. Amaranth's an interesting crop in and of itself. If you've never seen it, you're going to see it today because I hear we have some out there. that Kevin nicely planted for me, some populations. But here's quinoa, used as a pot herb. So once again, more back to our previous speak, used as cutting off the leaves and using it as a vegetable type process. The seed itself is also very nice. It's got a nice standard, it's got high lysine. Most of the cereals will have low lysine. It's a pretty balanced source, so pretty good source of nutrition. Also domesticated anciently by different ancient cultures, if you would. But once again, also was maybe, as Luz suggested, as Dr. Gomez suggested, was just something that got replaced by other crops that came in, the cereals like wheat and barley. But certainly it was very important. If you think about it, the Montezuma, the Emperor Montezuma, required equal tonnage of amaranth as he did of corn. It was that important to them in their culture there. So a pretty interesting crop in and of itself. It grows through these areas here, still fairly important in regional areas within Latin America. And of course, you can notice that there's at least 3 different species of the grains. They're used as grains in these areas here. And then there's this big wild weedy that you guys would think of as pig's weed. In fact, whenever I grow this, most farmers think, what are you doing planting weeds? I'm sure Frank's had the same situation there. But today you can still see these crops, very important in regional markets. This would have been in Michoacan in Mexico. So very important out there. I want to tell you guys a little bit. This is my plug for David Brenner. I don't see David. Where is he? David is our curator at USDA, right, for the Chenopodiaceae, or Amaranthaceae, I should say, really, as they changed its name. And you guys can see up here, we've got some stuff, but these are genera. So this isn't quinoa. Quinoa, we probably have maybe 300? Yeah, 300 accessions of quinoa publicly available, all brought in legally, available through David Brenner at the USDA in Ames, Iowa. You wanted to have that. And so, that seems impressive, Amaranth and Chenopodium, until you start thinking about what you should really have, like wheat. 70,000 of wheat, of not Chenopodiaceae, a whole bunch of genera, and then you get a few lines. This is maize and wheat out there. And so I still think we have a long ways to go. Even when we look at collections like the one that they have in Proimpa, the one we have in La Molina, while those are very big at 4,000, I still think there should be some effort made at making sure we don't lose the natural diversity that's out there. As we develop new varieties that are going to be higher yielding, and we want farmers to use those, I worry, and I'm sure that Lewis and others would worry about this, what happens to the material that was passed on from father to son over generations. We don't want to lose that. We want to make sure we capture that in our own germplasm banks, whether that be in the USDA or regional banks there. But we want to make sure we don't lose track of how important the USDA has been in their GRIN, in their germplasm system. So there's my plug for Dave. I wanted to make sure he got that. If you can't get money to work on these crops, be inventive. I sell this to USDA as a weed and then use all my efforts on the grain species. But, amaranth, here are the different amaranthus species. These are the weed species. And if you think about these, among the top 20 weeds worldwide, 6 of them are amaranths. And so it's easy to go about saying, hey, anything that I do in Amaranthus hypochondricus or Amaranthus coentus, these are the grain species, is directly applicable to these different species that are out there. In fact, we worry a lot now that we have these super weeds that are resistant to Roundup. In fact, the amaranths are unique in the sense that they mutate very rapidly, as does quinoa, and it's one of the few species that has overcome all the major herbicides that are out there. There's ALS resistance, there's PPO resistance, there's glyphosate resistant amaranths that are out there. So it's something we need to monitor as we go forward. It's also very interesting in that you'll notice that their sex strategies are unique here. You have dioecious, you have male and female plants, and then you have other species that are complete flowers where they're in the same plant. Interesting as well in these guys is you have some of these species are C4, some of them are C3. So within the same genera, we're seeing different species using different adaptations for photosynthesis. Very interesting to look at. Well, not to bore you guys, but just so you guys know, first thing we did when we got involved in this project as geneticists is we started making genetic tools. So there's a back library for both amaranth and quinoa, and they're pretty good. They're 10x. A back library is just a big chunks of DNA that have been characterized. And so that makes it easy for us to go in, take these big chunks of DNA, probe them for a very specific gene. So we're interested in herbicides. We're interested in the targets of these herbicides. One target of the herbicide, here's PPO, this is prototype porphyrogen oxidase. It's a herbicide like Blazer or Flexdor that attack this enzyme right here. And we know that this enzyme is mutated in Amaranth. We'd like to know what the mutation was so we can make sure, we trace that through populations. If we grew out Amaranth in bigger cities, we could actually monitor this gene for mutations to make sure we don't end up with herbicide-resistant amaranths that are out there. And you can go back here, you can just probe this back library and easily pull out the entire gene here, sequence it, and find the mutation that's involved in that. Fairly easy to do that. We've, of course, done that with the number of genes, glyphosate, ALS, PPO, all those target genes have been cloned out, and we understand how they're used quite efficiently now. We've also used the one in quinoa to pull out the salt resistance gene. This is one of the more famous. This is obviously a quantitative trait, salt resistance, salt tolerance, I should say. But one of the more famous genes is salt overly sensitive 1. We cloned that out, but probably more interesting to us from a basic standpoint is that quinoa, as it was said, is an ancient polyploid, meaning that 2 diploid plants came together, and now quinoa shares 2 copies of every gene. Okay, and so it's more difficult. That has implications on your breeding. Other polyploids, of course, you guys would know would be wheat and those types of things that are out there. But we were able then to sequence the 2 different genomes where the SOS1 and SOS1B genes are, and you'll notice that they're almost all identical. Those genes are very close. Those have implications when we do genetic markers and mapping stuff there. But we could also then look at this and determine how long ago these species diverged. And so the diploids diverged about 6.3 million years ago. And then they came back together to form, and Rick will talk about this tomorrow, then they came back together to form what we think of as tetraploid quinoa. But the diploids are very old in that sense. We also looked at the seed storage proteins. So one of the things that we know about quinoa is that the seed storage proteins are very important. They're highly nutritious. Well, what are they? Well, we've cloned those out, and those are easily to get at it now. We sequenced a lot of the genome with these back clones, and we were trying to look for interesting ideas behind it. And it turns out we knew there would be transposons and retrotransposons in there. We see quinoa, and it mutates, and you'll see sectors of leaves that are blue and sectors of the head that are red. That's often transposition of DNA transposons or retrotransposons. We find those, but then when we go to GenBank and we ask ourselves, do these things look like anything that's in Arabidopsis or anything else? No. Nobody's looked at these things. We know they're there. You see them phenotypically in the field. They're active, they're there, but when you go and try to find them in the genome, because we don't have an idea of what they look like, because nobody's worked in Chenopodiaceae or Amaranthaceae, we can't find them very easily. So there's a wide open field if anybody's interested in looking for the transposable elements that are in quinoa or amaranth. Just to go a little bit further, we started to think we wanted to develop genetic markers. The use of genetic markers, an example would be when Frank was talking about making crosses or having crosses between C. giganteum and C. quinoa, and thinking, well, I can look at the stem and find difference, but you'd like proof. Someone says, do you know that? Send me the materials. I can look at the genetic markers and say, this one had this allele, and this one had that allele, and this new thing you have has both. That's a hybrid, and that's definitively a hybrid. And so we were looking for those types of genetic markers to use those. And we, of course, picked those out, and they were very easy to find, to be honest with you. And I can show you guys these ones. There's some that are monomorphic, but there's others that are highly polymorphic, easy to use, and they're all available. The other thing I would note, this is Amaranth. And, of course, you can see they're useful. This is the same marker, and they're useful across lots of different species of the same genera there. And, of course, you can see that they segregate just as you would expect in an F2 population. Here are the ones for quinoa. You guys will notice that as well. Very nice. There's good genetic resources as far as genetic markers out there. I think there's about 400 SSR markers that my lab alone has developed and characterized and published in Crop Science, if anybody's interested in those. The other thing I'll notice on this one, this QCA57, you'll notice it amplifies not only one loci, but then another one here. This is the second genome that's being amplified at the same time. It also helps us figure out a little bit more about the lineage or the ancestry of these plants. Flipping back to Amaranth here. Remember, there's 3 different subspecies of Amaranths that are cultivated. In Mexico, it's Hypochondriacus. In Central America, it's cruentus, and in South America, it's caudatus. And when you use the genetic markers, look, you can separate those apart really easy. But if you throw in the hybrid, or excuse me, the hybridus, which is the weedy species, the pigweed, you'll notice that it falls out into all the different clades. It's actually not in this one, but it is in more data. And it turns out that hybridus is probably the potential parent, which makes sense, but that wasn't what people were thinking. They were thinking hybridus made cruentus, and that crossed with quintensis to make caudatus, which crossed with pallii to make. That's not what happened. And the markers showed us that it looks like these were independent domestication events. Some farmer in some field said, hey, this is interesting, this amaranth. And it was a hybridus at that time, and he selected for it and got out of that caudatus. And this farmer over here, in some independent time, selected out and probably got hypochondriacus out of that. But all coming out of hybridus, as you would expect in those cases. We have these genetic markers readily available. They're, next generation type markers, so they're fluorescent. You run them on a sequencing gel, easy to use. They're multiplex ready. In fact, Francisco Fuentes at Iquique in Chile has already used those to look at the Chilean germplasm that he had available. I would welcome any collaboration to look at your germplasm, whether it be in Bolivia or in Peru. We're starting those things now with, in fact, Jorge Jimenez Davalos is in our laboratory now. Actually, he's leaving yesterday. He left yesterday, and he's running the genetic markers on the materials, some of the materials that Luz has. We'd welcome anybody to come with their materials and leave with their materials and get the data. That way we avoid those issues. Am I out of time? Is that what you're telling me? Oh, Argentina, Michael. Oh yeah, and that's right. And Danielle's used them also in the Andean materials out of Argentina. Very nice paper there with the. Oh yeah. So stuff there. We've looked at the USDA material. We've compared the USDA material to what we have in the international nursery, which I should give thanks to Sven and Angel Mujica at SIP who put together this. I think I'm right here. You guys were partly responsible for picking this broad set of germplasm that was sent out and publicly usable. We used that in an experiment with our USDA material that we got from David to see whether the USDA material was encompassing or were we missing part of the germplasm that was in quinoa. And what we find out is that we've got most of it. We've got a lot of the material, and we cover in those 300 lines a lot of the diversity that's present, at least as recognized by that SIP international nursery that was out there. We would notice to you guys here, this set of materials that is right in here, this is in the GRIN system from a fellow named Emigdio Bayong, and it's probably hybrid material. So a good starting place if somebody wants to look for materials that might already be the result of hybridization between coastal lines that are over here and altiplano lines that range the geographical localities of Bolivia down to Peru and those areas there. Oh, sorry. Oh, it was underneath there. Sorry. Nobody looks at those things, right? You just email me and I'll get you the right things. We started making populations here. Here's one of our populations. This is a valley type out of Peru, 0654, and then this is a coastal line KU2. Very early, you guys can see some of the information here, the earliness of it, the size of the seed head, those types of things. And we started thinking we want to start mapping characteristics out of that. So we were making crosses early on, making genetic maps. This is our very first attempt at a genetic map for quinoa in 2004. It's AFLPs, completely useless because they're not transferable. But then we moved on to AFLPs, and I'll show you in a minute. We've moved on to SMPs. We have a map with about 500 SMPs on it now. So we're moving in the right direction. Here's Amaranth, if you've never seen it here. Here's a variety called Love Lies Bleeding, because it's got this nice bloody looking tail that's coming off of it there in fluorescence. Across to another one here. You guys can see very easily. It's easy to pick up the hybrids here. Once again, just from a morphological color, you can see it's segregating 3-to-1. These are nice proven F1s that are out there. We looked at them with our SSRs. So here's a cross. Here's what the F1 looks like. Here's a cross. Here's what the F1 looks like. We ran 73 microsatellites on those, and you guys can see, hey, this was not very polymorphic. Turns out if you do a little bit of research on this, you find out that these are actually sister lines, and so they should be very close to each other. And then we cross these ones, you get a lot more polymorphic. In fact, if you cross these ones, you do really good. In fact, you get so good that the lines start to mutate. They look stumpy, and something's wrong with them. And when we asked for this material from David, he'll appreciate this comment here, we said, send us some good material that we can plant in La Molina in Peru. So we wanted caudatus lines. So we said, David, we don't know a lot about Amaranth. Can you send us two things that look really, or a bunch of things that look really different that are caudatus? So he sent us all this material, and we made all of these crosses, and you guys are going to find out this is not caudatus. So be careful with what's in the USDA. Yeah, I know, but it looked different, right? That's what we told you to get us. It's hypochondriacus. And of course, that means if you cross the caudatus line with the hypochondriacus, you're going to get a lot of polymorphism, a lot of differences out of that. We decided that, hey, we need to move to the next generation of genetic markers. So the SSRs are great for phylogenetic type work, ancestral work, but for genetic mapping and for association mapping type studies, you really need the SNPs because they're so much easier to score. And so we started thinking, how are we going to come up with SNPs? We published this paper in 2009. We just did a quick genome digestion, and then we sequenced between the two parents of each one of our sets here. And you can see our two parents. And then we align them, and we look for a single nucleotide that's different, and that becomes our genetic marker. And these fairly straightforward to do this kind of stuff. I only present this because I knew that this would be kind of a mixed cloud. This is the newest method, or one of the new methods, next generation sequencing methods. This is a plate that's about yay big. So the size of a microscope slide, very small. And what you see here on each one of these lights is where they're doing the sequencing reaction. So we're almost doing all of the genotyping by sequencing these days, and they're being done on next generation sequencing. This little spot right here is this, and there it is there. Each one of these wells represents a different piece of DNA that's being sequenced. 5 years ago, you did a plate that had 96 wells in it. One of these lines right, one of these lines right here. And of course, today we're doing these types of things. I think genomics is really moving forward, and that means it's accessible because the cost is so low. It's accessible for any of these crops you start thinking about. Quinoa, very accessible. We certainly are sequencing, and we're going to move towards a full-blown sequence of Amaranth and quinoa very soon, I think. But you can align these things, and then you can look for these polymorphisms right here. See those? Once you do that, then you can start asking yourselves, well, do my SNPs agree with my SSRs? You guys can see very few SNPs were identified between these two. Very few SSRs were identified. You guys can see the same thing here. But all of a sudden, in these types of crosses, you can find 11,000 genetic markers on a single population and map those. And so this is of course, Amaranth in this case, but we've done this as well in quinoa, and I'll show you that map in just a minute. We also developed new technologies for genotyping. Doing it on gels, pretty painful. So we've been using this Caspar system, which is a closed system. All you do is do the PCR amplification. So Frank's interested in seeing if he's got a hybrid, cuts off a piece of leaf, I extract the DNA, and I just pipe it into a single tube. There's no gel running. There's no electrophoresis. It's all done, in a closed tube system with fluorescent dyes on them. And you guys can see here, it's very easy to pick out the hybrids, which would be in this area, and the homozygotes, which would be in the other areas. Nice thing, of course, about this is we're getting our data point costs down. But we've gone even further than that. We're now using the latest technology from Fluidigm. And again, come to the lab, use our equipment. The capital equipment's the expensive part. We've got it purchased. We'd love to share and utilize this with anybody that comes in. But in this case, all you do is you put your primers that are going to mark your SNPs or your genetic markers on one side and your genomic DNA on the other side. And then it mixes them all together. You can see these little channels. And so instead of mixing in different little tubes, this thing does it for you in an hour and a half. It makes 9,216 PCR reactions in an hour and a half. Get your graduate students to do that. Huh? Yeah, not as easy. So we think about it as a serious time saver, labor saver, reagent saver. And of course, data looks pretty good. Pretty nice to do it this way. And you can kind of see what data would look like. Here's an F2 population down here. And the blue are the heterozygotes. So remember, a SNP is this way, and then these are the F2 lines going this way. And so if you look at this, you would expect to see a lot of blue because these are F2s. You'd expect to see a 1 to 2 to 1 ratio, where the 2 would be blue, the heterozygotes. And then the red and the green would be the homozygotes. The other thing you might be able to see up here also is that these can be used on the different species as long as they're somewhat related. And I'll pull those up so you guys can see them. Here's caudatus. You can kind of see it even without them. They separate nicely into what you'd expect as bins: caudatus, cruentus, and hypochondriacus. And of course, you can make these very nice dense genetic maps very quickly. I had an undergraduate make this genetic map of some, I don't know how many, 480 markers. He did, an undergraduate did it in 2 months. I did my whole PhD in 4 years, and I put 136 markers on soybeans. So I'm pretty impressed with where we're headed, and I think it's getting even better. Here's some of the populations that you guys will actually look at. This is what they look like as an F2. You guys are going to see them in the field as an F7, I believe. So we have nice materials that can be utilized. I told you guys that we screwed up. Here's that one individual. It's supposed to be in with caudatus, but you guys can see it clearly is in with hypochondriacus. These are things that you can figure out with the genetic markers, which would otherwise be hard to see just morphologically. Certainly you can prove those things to yourself. The other thing we proved to ourselves as well is that hybridus was the event to caudatus. Hybridus was the event to hypochondriacus. Hybridus was the event to cruentus. Individual selections by farmers in these different cultures leading to these different ones there. And of course, this is the information that's just published now in 2013 by Jimenez in Crop Science, looking at amaranths throughout Peru there. Our cross here, the genetic map that came out of quinoa, the same technology. It was a recombinant inbred line, so you only expect the homozygotes that are going to be in there. So very nice technology. We invite anybody who wants to come to the laboratory to use that. You can also clearly use this to separate out the different lines. These are the coastal lines. These are the altiplano lines and some of the valley lines. I should have marked them out better for you guys, but I didn't. One of the things that you'll note out of this is that the coastal lines are tremendously unique from the altiplano and valley lines, which tend to group back together. And I think I'm going to spread rumors. Who was it that said I shouldn't do that? Pablo told me I shouldn't be spreading rumors, but what I've been thinking about in this one is that, you know, these people, the Indians there, that Mapuche tribe, the Indians that were predominated this area of southern Chile, were never conquered by the Incan, and they had a restricted gene flow between their material of their quinoas, which they called quinoa, and the quinoas that were then mixed, and you really did have this intertrading amongst the different Incas at different levels in the, you know, at the coast and everything. So it's a much more jumble up here, whereas you don't see that here. It's very much a genetic bottleneck that occurred in this stuff, and you easily pick that up with the genetic materials up there. Yeah, it's potentially, when you cross it, you get mutants. Anyways, we also, just so I make sure I can stay a little bit on time here, we've been working on RNA-seq and EST libraries. We wanted to find the genes, for those of you who are not genomics people. We wanted to try to pull out all the genes so we had ready access if somebody wanted to go look for drought resistance or salt resistance, those types of things. So we set up a number of experiments with quinoas. In this case, we used Ingapirca and Oyagüe. One's a salt tolerant, one's are drought tolerant, and one's more drought susceptible. One's from Peru, one's from I think the altiplano on the Chilean side of the altiplano. But you stress these guys out at filled capacity, so 100% filled capacity would be over here, 50%, 30%, and then 10%. And you start to see things like stomatoconduct and stem water product. These things are uniquely different on how they control their water potential. We want to understand what genes are being turned on. So in a replicated manner, we take all the RNA, the mRNA, all the genes that are turned on in these different treatments, in a replicated, I think that is important here, any of these experiments, replicated in 3 blocks in a randomized complete block design, and then sequenced what genes were turned on at these different levels. And you can see we're sequencing 385 million fragments to look at this, to try to get a good sense of what's being turned on, what's not being turned on there. And what you come up with is there's about 20,000 genes that you can look at, and you can get some good ideas of what they're involved in. And then you can say, what's the difference? What genes were turned on at treatment 4, but not at treatment 3? And then you can get these lists that start making sense to you. Oh, here's one. You know, this gene, I don't know what to call it yet, but it's like a serine threonine protein kinase. And of course, it's the main effector in hydritic stress response in Arabidopsis. And it shows up in our analysis. So that list of genes is publicly available, as is the DNA sequence. One of the things that at BYU, we made an early on obligation with our work with ProIMPA in Bolivia that anything we did, we'd publicly make available. And so as soon as we get it, we try to open it up to anybody to use markers, DNA sequence, and those types of things. So there's a nice EST library, cDNA library, if you would, that's available out there. Just some of our collaborators that have been out there. It was nice to come last year and actually see some of this stuff in an agricultural setting. Kind of excited to talk to some of the rest of you guys that are actually doing this in a much bigger way as well, and come see your plots there and some of the stuff that's out there. you know, combining. This is what our fields look like today. This is our replicated field trials of the Amaranth that we'll see there. You guys can see what it looks like out there. Here's what it looks like apparently here at Washington. I haven't looked at your guys' fields yet, but that looks pretty good to me. I'm excited to go see that. This is some materials that Luce has provided to us. This is the non-mutated line, and here's her dwarf lines. We got a whole roll of these things that come down. So some great materials that are out there, making the right types of friendships and being honest and trustworthy with these people have gone a long way for our group. Rick and I are very thankful for everybody that's worked with us, from Daniel, Sven, to Luz, to Alejandro and Willie. Just, it's been an awesome opportunity to work with these people, and I can't say enough. What I think we're really headed towards, you guys, in a genomic sense is, you know, I think we're going to get the sequence. I don't think I'll get that to go, but we've been sequencing back clones, and this was going to show you all the sequence for about a minute. It would make no sense to you. But that's exciting because it's an empty box of sequences that we're getting, millions and millions and millions of sequence, and we're trying to figure out what it all means. But definitely with the slope of sequencing a genome dropping now to $10,000, we're going to see sequences of many of these species. And we're going to need people to start looking at the genetics and start to apply that to the agronomics. And I think we're finally seeing with groups like Kevin, and that's why we're so excited when he called. I'm sure he was like totally like, what's wrong with these guys? They're so excited to collaborate. We're doing all of this genomics work, and we had no way to apply it to a field setting. So we were excited to see this all coming together, and we think there's even more coming. Come visit us in Utah. We managed to get a couple of scientists. This is a scientist from Nicaragua. No, where's Mario? El Salvador, and then of course Luz, and then Elogio from Mexico, and we'll bring you down to what a real environment looks like, you know, those type of things. A lot of funding agencies out there, and I'll leave it at that.